Today we're going to talk about staining. And what we're doing is we're trying to make the bacteria stand out against the background. So we're putting a dye onto something in the cellular structure to give it contrast. We will use mostly basic dyes like methylene blue, crystal violet, safranin. Some of them you used yesterday or the other day in your simple staining because they are attracted to the negatively charged components of the cell, which is the cell wall, which helped us give the idea of what shape and arrangement the bacteria happen to be. There's also acidic or anionic dyes that are attracted to the positively charged parts. A simple stain is what we did in our last class where we used a single dye and it revealed the shape and arrangement of the bacteria. A differential stain makes use of two or more dyes and it will actually distinguish or separate the bacteria based on structure. The Gram stain is one of the big ones that we will do.
The Gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the Gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of Gram's iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the Gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
So the basic steps for the Gram stain are you're going to make the smear, which we made the other day because for a Gram stain it needs to be 24 hours old, and you've already heat fixed it. The very first stain or primary stain is crystal violet, and we'll do it like the simple smear in that you flood the slide and let it set for a minute, and then you can drain it or rinse it with water. Something new is iodine. Iodine acts as a mordant, which actually binds to the crystal violet and will keep it inside the cell wall. And you let that set on the smear for a minute. Crystal violet and iodine, make sure when you put them on, they haven't weakened and they're not too light. If they are, just let me know and we'll replace it. The decolorizing step comes after the iodine, and that is our ethyl alcohol. And I like how the video explained drop by drop, because this is one of the steps that you can ruin the slide's actual end results if we over decolorize it. So basically do it till it runs clear, and then you can rinse it with water. Rinsing with water doesn't hurt at any point in time in the gram stain. And then our counter stain will be saffronate. And to make our lives easier, I say 1 minute here, and then again you rinse it and blot it dry with our bibulous paper, and we look at it under the scope. This is a great way to remember the order: come in and stain. It's crystal violet first, then iodine, then alcohol, and then saffronate. Positive and gram-negative cells. Positive will be purple under the microscope, and gram-negative, since it did not retain the crystal violet, will be a pink or reddish color. Okay, again, you make your smear, you let it air dry, you heat fix it. Crystal violet is our first dye. You rinse it, then you use iodine, which is the mordant, to help hold the crystal violet in the cell. You rinse it with ethyl alcohol, and then you counter stain with saffronate. Again, we're going to have to use immersion oil to look at it under the scope. Okay, so with the fixation, the first dye, it's always going to be purple. We can't tell the difference. Then you add the iodine. The decolorization is really the step that will help distinguish gram-positive versus gram-negative. Keep in mind, the saffronate is staining the gram-positive, but it's so purple we can't see the different color. There's a Quizlet for you if you want to review the steps in the gram stain, because you will. On Tuesday, we'll come in and we'll be ready to gram stain the other bacteria or the other smear that you make.